The ocean is like a universe to be discovered.
This universe includes algae, both macro and microalgae, which have developed powerful defenses against environmental stresses. Their tools may also benefit us.
This algae world is like a huge untapped reservoir of potential compounds that it's like infinity.
With the help of these algae, scientists in the Algae for IBD project are tackling inflammatory bowel disease, IBD for short. IBD is a chronic disease that can be triggered by lifestyle, diet, and stress. It affects millions of people worldwide and is a lifelong condition.
It's a chronic inflammatory disorder of the gastrointestinal tract that interests the whole tract from the mouth to the anus.
Reoccurring inflammation damages the digestive organs, leading to severe pain, exhaustion, and, in some cases, even colon cancer.
There is no effective cure, so these patients have to deal with either surgery or steroid-based treatments or something that they can relieve their symptoms and pain.
Today, the number of people suffering from chronic inflammatory diseases is increasing, with almost 6.5 million people affected by IBD alone. For these patients, everyday activities such as eating, working, and travelling can be unpredictable. Although IBD treatments exist, many people repeatedly but unpredictably experience sudden resurgence of the inflammation.
Fortunately, today we have several drugs, but these therapies are costly, and frequently patients have a relapse of the disease during the course of therapy.
Therefore, new anti-inflammatory compounds are needed. This is precisely where marine biology meets clinical science, and the European Algae for IBD project began.
Microalgae have a very good amount of protein. Some strains can even reach like 70% of protein, which is much higher than meat. They are also rich in the omega-3 fatty acids that in our food we get from fish. And at the same time, they are rich in all sorts of bioactive compounds, so secondary metabolites that have particularly interest to our health.
In addition to their vast variety of possibly beneficial compounds, algae can also be grown efficiently and sustainably.
Microalgae are photoautotrophic. They use solar energy to produce biomass. They also use CO2. Microalgae can also grow in seawater or even in wastewater. And we are not using arable land because we don't need the land to grow microalgae. We can grow microalgae in the desert if needed.
First, the Algae for IBD partners compiled a diverse collection of over 1,000 algae strains from existing algae libraries. They then narrowed this collection down to 150 promising strains for dedicated research. The next step was to cultivate these algae on an industrial scale, using advanced indoor and outdoor systems.
Each one has its own specific needs, and we provide those needs. So we adapt to the algae. Some algae like a higher pH than others. Some don't like the speed of the reactors. And we also try to adapt the algae to us. It's like a meet me halfway strategy.
Some molecules that are already available to help patients with IBD are difficult or expensive to produce synthetically. This is one of the reasons why algae are being considered as a natural, sustainable source of rare bioactives containing anti-inflammatory and antioxidant compounds. Even when an extract shows strong activity, the research work is far from done. The scientists need to understand the 3D chemical structure of the active molecule, for which they need to get the molecule in pure form.
To get the compound in a pure form so that it can be chemically characterized, it can take us 1 year and a half, sometimes longer. So the compounds can be present in the biomass in such a small amount that the purification process is extremely complicated.
Here in Milan, the potential of these algae-derived compounds was tested on real, inflamed human cells to see if inflammation-related signals decreased.
We take advantage of the mucosal biopsies. So we can collect mucosal biopsies from the intestine of patients that undergo endoscopic surveillance during the check that they have for their disease. And the biopsy that have been collected can be put in a plate and then treated with algae-derived compounds.
55 IBD patients underwent endoscopy and provided biopsies for the lab test. These tissues were cultured and exposed to algae extracts.
We can measure after 24 hours, 48 hours of treatment whether they have been effective. The algae-derived compounds can exert anti-inflammatory properties. They are quite tolerated from the human organism, and so they can be used as an alternative therapy for IBD.
To make these bioactive compounds available to IBD patients to support their daily lives and gut health alongside standard care, the project also aimed to develop tasty, functional food.
IBD patients are lacking effective treatments. So one of the project aims is to provide them with active functional foods based on those active algae that can support their immune system, and they are not a replacement for their ongoing treatment, just to support their other aspect of the disease and symptoms.
2 industrial project partners in France and Ireland already started to integrate those algae compounds into daily food products.
We are an industrial partner responsible for the scale-up of the production, but also for food prototypes, meaning that we want to bring solutions to IBD patients in the forms of gummy bears or cereal products or dairy products.
As well as functional foods, the scientists also want to develop a new drug. However, the drug development process is slower because it requires many preclinical and clinical steps. Nevertheless, Algae for IBD is designed so that each step builds on the knowledge gained in previous stages.
It is challenging to produce a drug for IBD. Probably thanks to preclinical studies, we can verify the efficacy of these drugs.
While the Algae for IBD project ends, research on algae and the search for beneficial compounds derived from algae are just at their beginning.